So first of all, thank you for joining this webinar. I will present to you some of the work that we do at the biological lab of the Belgian Defense, and specifically a project on the detection of biological toxins. So it is called RICIn detection, identification, and verification of the biological activity, RICIDIV for short. So the agenda for today will be as such. First, I will give you some background on the RICIn toxin and on the project. Then we will dive into the large-scale production of reference material that we developed during this project. We will then look at our new method for monoclonal antibody production against ricin. And as a for our fourth section, we will look at the ricin detection itself. So in particular, immunoassays that we developed. And finally, we will look at the outcomes and the perspectives of this project and our toxin section about the LDBO in general. So what is RICIn? RICIn is an ever-growing threat. So it's one of the main potential biowarfare agent. So this was showcased in 2018 with the colony terrorist plot in Germany, when an individual built a makeshift clandestine lab to produce large quantities of ricin. So he produced enough ricin to potentially kill hundreds of people. Thankfully, this terrorist plot was foiled by the German police and the German defense, but it showcases how relevant it is to have methods in place to safeguard us from potential ricin attacks. So what makes ricin such a potent biowarfare agent? So several things. First, of course, its lethality. So it's highly lethal. Several micrograms of ricin could be enough to kill you. So there's different administration routes. So ingestion, injection, and inhalation. With injection and inhalation, less than a milligram is enough to kill you. With ingestion, higher quantities are enough. It can be lethal, but at much higher quantities in hundreds of milligrams. The ricin itself, so first of all, so as I mentioned, its lethality. Second of all, the availability. So ricin comes from a plant, the Ricinus communis plant, which is a very common plant that you can find in gardens in Brussels, for example. So it's very common and easy to find. And third, how easy it is to extract the toxin from the leaves. So you don't have to be a highly trained chemist to be able to extract this toxin to then use as a biowarfare agent. So the ricin itself is a dimeric protein. So you have here, I will add the pointer. You have here the A chain and the B chain, so dimeric protein with two chains that are bound together by a disulfide bond. Its method of action in the cell. So you have the B chain that binds onto the cell wall, which facilitates endocytosis of the entire protein. So as you can see here, the A and B chain enter the cell. The ricin travels to the endoplasmic reticulum here, where the two chains are separated, and the A chain is released in the cytosol. The A chain can then inactivate translation by binding onto the ribosome, which effectively kills the cell. One ricin molecule is enough to kill a cell. So this is why the Belgian Defense started this project to generate and design a multi-assay pipeline for the detection and identification of ricin. So here you can see the strategy we put in place. So first, the biotech production of molecules, which entails producing ricin and producing an inactivated version of ricin as well to be able to work safely with it and to be able to develop new methods of detection and identification of ricin. Second was the production of antibodies using the ricin that we generated in the first part. Using those antibodies, we can then develop subsequent immunoassays as shown here. So the development and optimization of biomolecular assays, specifically lateral flow assays and ELISA assays that I will go into more details later in the presentation. We also have here in the third step the optimization of LC-MS, so liquid chromatography coupled with mass spec, which is the gold standard for chemical warfare agents, so CWA detection. This we do in tandem with the chemical analysis lab at DLD. So there's several steps that you can optimize before the LC-MS itself, namely the enrichment of the antigen within a matrix. So if a matrix has a very low quantity of the target's toxin, or if the matrix is a complicated matrix to work with, you might need to enrich it. So increase the amount of RICIn in it before you can detect it. And also the digestion of the protein itself. So with LC-MS, you actually detect peptides, so small fragments of the entire protein. So before you can actually detect it, you have to digest the protein and divide it into many different small peptides. So after those two techniques, there's also the bioassays, so the functional bioassays, I can call them as well, which is the assessment of the toxicity of the RICIn. So for example, with cytotoxicity assays, you put the RICIn in contact with the suspected samples with potential RICIn in it, in contact with a eukaryotic cell line, and you determine if the cell line survives, or if there's actually cytotoxic activity within the sample. And finally, with all those assays, you validate it in the lab and afterwards in the future also on the field, giving us an integrative analysis pipeline of RICIn. So first, the production of recombinant RICIn. So I first need to give you some background into recombinant production in general. So this is a method that we use and biologists in general use to produce compounds. So here to produce proteins using live organisms. So here we use E. coli, which is the standard prokaryotic organism, so bacteria, and we add our gene of interest. So here we have our RICIn gene. We add it to the bacteria here on a plasmid, so a circular DNA structure. We add our target gene, and then our protein will be expressed by our E. coli bacteria. So here we have the production of our different chains. So the RICIn A chain here, the RICIn B chain here, and the RIVAX chain, which is an inactivated version of the RICIn A chain, so with no residual toxicity. One important point is when the two chains are separate, they are not lethal, they're not toxic, because the RICIn chain needs, so the toxin needs the B chain to enter the cell, and then it needs the A chain to actually inactivate the ribosome. So one without the other are not lethal, but there can be some residual toxicity from the A chain. So this is why we also produced a RIVAX with absolutely no residual toxicity. This work was a collaboration with the University of Ghent and specifically the Center for Synthetic Biology that are experts in protein production. So here we have our purification. So this is a Western blot result where we have our free chains after purification. So as we can see, we have almost no background, so a high level of purification. We optimize the expression itself by fine-tuning different parameters. So on the plasmid itself, we can play with different sections of the plasmid, so the promoter, the gene copy number, and also the ribosome binding site to increase or decrease expression to increase the production afterwards of our chains. So we fine-tune the expression like this, but also by playing with the actual expression conditions, so the temperature, the concentration of the different reagents, and the length of the expression, for example. So you can then upscale it to 250 milliliter flask and keep on optimizing before doing the subsequent purification. So here you can see you have those histidine tags that we also add after our RICIn gene. Those tags are used to purify your protein after the expression by doing affinity chromatography. So you run your protein of interest after expression on a column, and the protein with his tags will bind onto the column, and all the rest will be eluted through. So that's how you purify your protein. As I mentioned, you can further optimize it with changing the temperature, the medium, but also adding chaperones, which will target your protein to the right location for increased expression. So after this initial optimization, we did large-scale production of the RICIn A chain in particular. So here we have our optimized microbial cell factories, and we went on to 25-liter batch reactors for large-scale production. Here are the downstream processes afterwards, so the lysis of the cell to break the cell wall to produce our antibodies, the affinity chromatography, which is using the histidine tags, as I mentioned, and also size exclusion chromatography, which is a purification based on the size of your protein, and finally a buffer exchange to end up in the correct conservation buffer for our protein. So here we have our chains, which can be used for our immunoassays, but also for immunoenrichment. As I mentioned, when there's a low quantity in a matrix, you have to potentially enrich your sample in RICIn, increase the quantity of RICIn in your sample. And for RICIn A specifically, we used it to produce antibodies afterwards. These are the results of the purification of recombinant RICIn A. So here you have a chromatograph, with the purified RICIn A after the 2 purifications. And here you have an SDS page where you can see the different steps of the purification. And as you can see here, compared to here, there's much less background, which shows the purification steps. So the next step was the production of antibodies. So as I mentioned, this was a collaboration with a K11 spin-off called Pharmabs. So the MabMind platform is an alternative method of producing antibodies. So we used mice in this case to generate antibodies against RICIn A. And in this MabMind platform, you use 2 different techniques to generate highly specific monoclonal antibodies. So first, you use fluorescent activated cell sorting to specifically take the B cells that react against your antibodies, and you follow that with Sanger sequencing to have the exact coding sequences against that code for the antibodies against your antigen of interest. And finally, you perform recombinant antibody production in your eukaryotic cells. So to put this in perspective, I will show you the traditional hybridoma method. So here on the left, you can see the traditional hybridoma technique, and on the right, you can see our novel recombinant production method, so the MabMind platform. So the traditional hybridoma technique is the method usually used to generate monoclonal antibodies. So you start off with an immunization of the mice. You generate B cells that can produce antibodies against your target of interest, so against RICIn in this case. Those B cells are short-lived, so they cannot survive long in a culture. So you then have to fuse them with an immortal cell line, so myeloma cells here, which gives you hybridomas, so clones that are half myeloma, half B cells that are immortal and can produce your antibody of interest. So you select those hybridomas, you screen for the ones of interest, and you get your monoclonal antibodies. Some of the disadvantages of this method is low fusion efficiency, lack of stability of the hybridomas, and the difficulty and how expensive it is to maintain in culture. So this is also why we used an alternative method, and we developed an alternative method. which, as I mentioned, starts off also with an immunization of the mice, and is followed by single cell sorting of the B cells of interest, followed by sequencing of the coding regions, and followed by recombinant production in eukaryotic cells to produce also monoclonal antibodies. The advantages of this new method is after optimization, it can be cheaper, faster, and more importantly for us, you have an increased reproducibility and control. Because once you have the sequences, you can very easily reproduce them exactly how they are. So you don't have a loss of stability like you can have with the hybridomas, and it's easy to also share with partners and to always have the same thing when you produce it. So with this method, we obtained a panel of 8 suitable RICIn antibodies that we used in immunoassays. So first, the sandwich ELISA. What is a sandwich ELISA? So here you have a schematic of a typical sandwich ELISA. So you have a capture antibody coated on a plate. You have your antigen here, so RICIn, and you have a second antibody, so the detection antibody that binds onto the RICIn. So the second antibody, the detection antibody, is beforehand it is biotinylated. So there's a biotin chain that is added to it, and this biotin chain will allow a reaction with streptavidin HRP and a subsequent substrate to react and to change the color of your solution. So if RICIn is present in the sample, you will have a sandwich, a capture antibody, RICIn, a secondary antibody, and a reaction that occurs. So here you can see a plate with different concentrations of RICIn going from up to down, also tested with different antibodies. So here you see different types of antibodies, and here you have the different concentrations of RICIn, and you can see a gradient as you go down in concentration, showing the reaction and the presence of RICIn in the sample. So some of the results we obtained by sandwich ELISAs. So here you have two experiments with our panel of antibodies. Here you have 5 of the antibodies from our panel, B1, B11, C2, D1, and D6, working in a sandwich ELISA in tandem with the R109 antibody, which is a control RICIn, anti-RICIn B antibody from the Robert Koch Institute in Germany. And we also have here the control antibody against RICIn A, which is also from the Robert Koch Institute. We can see from the results that the sensitivity of our antibodies in tandem with the R109 antibody is comparable or better compared to the control R18 antibody. So here you have detection of RICIn from 100 nanograms to 0.1 nanograms. And we can still see some here at 0.01 nanograms for 2 of our antibodies. Here you have the same experiment with another control antibody, so the BRIC20 antibody from the CEA group in France, and you can also see a stronger reaction for at least 2 of our antibodies, the B1 and the B11 antibodies, and all the way to 1 nanogram you still have detection, and for our 2 main antibodies you also have sensitivity all the way down to 0.1. 0.01 nanograms per milliliter, 10 picogram per milliliter. So the sensitivity is comparable to the reference ELISA under these conditions. So a second thing we wanted to test was if we could discriminate between detection of RICIn and its RCA120 isoform. RCA120 is a tetramer, so with 2 A chains and 2 B chains, that is also present in RICIn, but much less lethal, so 50 times less lethal than RICIn. So we want to be able to discriminate between the two if we have a suspicious sample. So here, same setup as before, a sandwich ELISA with our panel of antibodies, so B1, B11, C2, D1, and D6, and tandem with the R109 antibody from the Robert Koch Institute. Here we can see that detection of RICIn is twice as much as detection of RCA120 for 10 nanograms per milliliter. It's about twice as much for all of our samples except D1, and as you can see, it's comparable with the R18 difference, so with our control antibody. So the relative specificity to RICIn as opposed to RCA 120 is comparable to the reference ELISA under these conditions. So the second type of the immunoassays we are developing is the lateral flow assays. So what are lateral flow assays? Here you can see a typical lateral flow assay after assembly. So lateral flow assay, all of you will be familiar with the COVID test strip that we used during the COVID crisis. So you have those membranes. So here you have a schematic of the membrane that you assemble. And at the end, you put them in a cassette, and you can use those lateral flow assays on the field for rapid detection of a particular antigen. So the setup that we have here, so you have first a conjugate pad with the reagents for the reaction. So you have all the necessary reagent and antibodies here. And then on the test strip itself, you have one line with RICIn A antibodies that are bound to the membrane. You have a second line with RICIn B antibodies, and finally you have a control line with anti-BSA antibodies, which is just a control, which then the control is also present in the conjugate pack. So then when you add the sample with RICIn, the sample will react, will go onto the conjugate pad, and once you add the working solution by capillary force, or the reagents and the sample will go down on the membrane, where we'll first react with the RICIn A antibodies, then with the RICIn B antibodies, and finally the control line. If RICIn is present, you will have both lines. Both lines will be present as well as the control line. If you only have the control line, you can say the sample is negative, at least lower than the limit of detection of our lateral flow assays. And if no lines are present, then the lateral flow assay did not work. The benefits and advantages of this technique is that it is quick, easy to put in place, and easy to form personnel to do it. But one of the disadvantages that has a lower limit of detection compared to reach-back confirmation methods like ELISAs. But it's the method that we generally use on the field for quick confirmation of the presence of a target toxin. So this is the device that we use to develop our in-house ELISAs, so the Cyflex Area S3, which is an ultra-low volume spotter. So finally, we have the laboratory validation of the RICIn pipeline. As I mentioned, we have different methods that we develop or that we are using for the detection of RICIn. So we have our immunoassay with LFAs and sandwich ELISAs. We also have our LCM-SMS with the chemistry analysis lab and the functional assays. So the laboratory validation involves taking different matrices. So here we have water, food matrix, for example, milk, and plant seed extract, spiking them with our target toxins, so with RICIn, the RICIn A chain, the RICIn B chain, an inactive version of RICIn, also RCA 20. And we spike our matrices, and then we test the entire pipeline on it. We identify methods with a strong potential for further optimization and implementation. The outcomes and perspective of this project. So as I showed, we have high quality reference material with novel production methods. So we have our microbial cell factories, produced our recombinant RICIn chains. We have the MapMine platform where we were able to generate and produce novel monoclonal antibodies against RICIn. And we have a multi-assay pipeline for RICIn detection with the immunoassays, LFAs, ELISAs, also the optimized LCM-SMS, and the functional bioassays. So this was a project with different partners. So you have the University of Ghent, Pharmabs, K11 spin-off, and the Belgian Defense Laboratories, and all of this was funded by the Royal Higher Institute for Defense. Thank you.